Okay students, so welcome to biomolecular spectroscopy course, and we are moving more or less towards the end of the course. So, this week probably is going to be last week. So, you see in this 7 week we have studied UV visible. I showed you lots of application of UV visible. We have studied fluorescence, where we can use fluorescence. We have studied FTIR, we have studied Raman, we have studied CD, an extensive lecture on CD, right. So, with this now already we have discussed many of these techniques and where we are going to use these in our research. In the last week, I want to keep it as a bonus for some of you who aspire to work in industry. You know, this spectroscopic technique are extremely important in biopharma industry, and this week is I will be focusing on how you can use the spectroscopy in industry, and in this today and in the next lecture we are also going to look at how we can use this spectroscopy in biopharma setting. So, we are going to look at these different techniques. So, now you have studied at least 4 or 5 techniques, and then there are many other techniques. Question comes, how to choose a technique, right? There are multiple choice you have, and some techniques is available with your industry, some you may not, and you may need to answer a question, right. So, in this case, when you are solving a problem, more than technique it is important what questions you are addressing and what answer you are looking for. So, before choosing a technique, answer the 3 questions. What property do I want to measure? For a biomolecule, do I want to measure a concentration? I want to identify the molecule like a chemical identity, or I want to determine the secondary structure or tertiary structure, or do I need to measure the microenvironment or dynamics of this. So, that these are some of the questions or property that you want to address. Then the question comes, how my sample is looking like, what is my sample? To understanding the sample is extremely crucial. Is it a protein, is it a peptide or a small molecule? What is that we are looking at? Is it an aqueous buffer or a solid? Is it a native protein or denatured protein? Does it have a low concentration, like a micromolar, millimolar, nanomolar? What kind of concentration do you have? So, what matters is the sensitivity, how much structural detail I want to understand, and what kind of chemical specificity I want to do that. So, 3 different technique, right, these different spectroscopic technique, 3 that we can discuss, or 4, UV visible, fluorescence, CD, FTIR, Raman, all 5. Let us see. So, different spectroscopic technique can answer different questions, and no one is going to replace others, all has to stay here. They are complementary, not a competitive technique. So, do not stick to any technique, use them wherever, whenever they are needed. So, let us draw a decision map, how to choose a technique. A decision map, how much is there? First question, how much protein I have or how much peptide I have to do the experiment. If it is low, or if I want to measure how much is there, the UV visible, you have to measure the concentration using the UV visible. If I want to address the question, is the environment changing subtle, then environment, like you know, the fluorophores are very sensitive to environment. In those case, we are going to use a fluorescence. If I want to understand the chemical parameter, like a chemical bond or a fingerprint, the functional group, then Raman or IR is the better technique. If I have to understand the secondary structure, and we want to look at the if secondary structure is preserved or not, then circular dichroism. If I want to analyze in water through packaging of those material, we can use in those case Raman spectroscopy. So, look at here, to address the different questions that we may have, we are using different techniques: UV visible, fluorescence, IR or Raman, CD and Raman. That is the things we are doing it. So, how much and why to choose a technique? So, technique by technique, let us go ahead and then we will do some case study here. Starting with simple technique, it is a workhorse technique, it is a simple, extremely simple technique, it is a quantitative. What it measures, just to refresh your memory, it measures the electronic absorption of a chromophore, that is the aromatic amino acids, those are chromophore, tyrosine, tryptophan, phenylalanine or the conjugated system, that is what it measures. It is used for determining the concentration, it also checks the purity or batch to batch consistency is to measure. The readout is for protein we measure at 260, where absorption happens for tryptophan and tyrosine. For nucleic acid it is a 260, where basage absorbs. The small molecule can be compound specific lambda max. So, that is what readouts are, and merits it is a sample technique, cheaper technique, it is a fast, highly quantitative, and you can recover all of your protein, that is a non-destructive. So, limitation is we really get a minimal structural information, right, and it cannot distinguish whether my protein is folded or unfolded, just gives you the concentration measurement of the protein. So, suppose you are working with a company which is developing the biosimilars, a biosimilar monoclonal antibody. In those case, there are two things: drug substance and drug product. Drug substance is actual drug molecule, and drug product is when you mix with excipients and optimize the buffer, optimize the stability, ionic strength and all those, that becomes drug product. So, DS drug substance, DP is drug product, right. So, when we are working with a biosimilar mAb, this drug substance release testing is done by measuring the UV visible at 280, that gives the concentration of the monoclonal antibody. And same extinction coefficient can be used across batch to batch to understand the dose accuracy, because you cannot play with a dose. So, in each vial or each syringe you have to measure the dose, and here you can have a quantitative simpler technique for batch to batch consistency. And now FDA, the Food and Drug Agency, expect UV visible as a primary quantitative tool. This is the basic minimum requirement that any agency can have to show that batch to batch consistency is there. Now, another one, fluorescence, it is a ultra sensitive technique, it is a environment sensitive technique. What we measure is emission from excited fluorophore, whether it is coming from intrinsic fluorophore or extrinsic fluorophore, that is what we measure. Basically, it is used for protein folding, protein unfolding, it is a conformational change. We can study the ligand binding, the aggregation onset, very important. The protein therapeutics tends to aggregate, and those can be very dangerous. So, fluorescence is a technique that can help you in understanding this aggregation onset. What we read is a fluorescence emission. We can use intrinsic fluorescence like a tryptophan fluorescence, or extrinsic fluorescence like ANS. You remember ANS binds to exposed hydrophobic surface area, thioflavin binds to aggregated sample and Nile red, all of these are extrinsic fluorophore that can be used. Merit is it is a very high sensitive, detects even subtle change that happens before aggregation, and it is used for excellent kinetics measurement. The limitation is we had discussed this inner filter effect, and it is a fluorescence specific and not quantitative for concentration dependent. It gives you a structural insight, but not quantitation. Now, one can use for aggregation detection in formulation screening. So, which formulation is very suitable for your drug product development, that you can use fluorescence based technique for formulation screening. Now, during the formulation development, you can like have a two formulation composition. You can look, that can be looked by identical by say UV and SEC. You can use orthogonal technique, size exclusion chromatography, to find it out which formulation is good. Now, fluorescence can detect early tertiary structure perturbation happens. If suppose you are, for a discussion, let us take you are doing in a tris buffer or Hips buffer, are these two buffer changing the structure of a protein? How do you know, right? Fluorescence can give you if the structure, tertiary structure is changing. So, you can see the red shift of tryptophan emission indicates exposure to the solvent, and this technique fluorescence allows to eliminate the unstable buffer before costly stability study. So, early on in your development for drug product, fluorescence can be invoked to look at what formulation buffer should be used. The third technique, CD, circular dichromosome, it is a gold standard technique for secondary structure, right. So, protein first state of folding happens by secondary, then it goes to tertiary. Now, CD is the good technique for measuring the secondary structure. It measures, as we have seen last week, differential absorption of left and right handed circularly polarized light. It is used for quantitation of alpha helix, beta sheet, secondary structure composition, also thermal unfolding, right. So, the thermal unfolding gives the stability of a protein. What we measure, the far UV 190 to 250 and near UV for tertiary structure 250 to 320, where aromatics absorbs and give you information about tertiary structure. Meritage, it is a directly a structural readout, you get a direct quantitation of a structural elements present in the protein, whether it is alpha helix, beta sheet, random coil, what kind of secondary structure it has. It is a sensitive to the folding change, and it is a widely accepted technique by any regulatory agency, regulators widely accept it. What we need, limitations are, it requires a clean buffer which does not absorb, so low absorbance and lower sensitivity than fluorescence, and here we do not get residue level details, you can get the overall fold, but not residue level. In biosimilar compatibility analysis, in a biosimilar versus reference listed drug RLD, you can have the comparison, you just record a far UV CD, overlay the two spectra, and same minima you should have a 208 or 222 that reports the same helical content. So, it gives you key evidence for secondary structure similarity CD, and if the spectra is overlapping, suppose here is my spectra and you can have like a dotted line spectra just overlapping on top of each other. This is RLD and your drug product, it says that my protein is same or it has same secondary structure like the reference product, and that is a biosimilarity test one can have. Then moving to the third technique, infrared IR or FTIR, it is a bond specific structural information we get. So, what it exploits, essentially it exploits the vibrational mode of chemical bond. It measures the secondary structure of amide 1, right. If you remember what was amide 1, amide 2, CONH, that is what we were measuring in the FTIR amide 1, and basically secondary structure you get a peak and where you deconvolute to get alpha helix, beta sheet, random coil concentration. So, that is what we do. If it is not, then solid state analysis we do. It also can measure excipient protein interaction. So, we read out is this wave like wave number 1600 to 1700 for amide 1, amide 2 1500 to 1600, and one can essentially deconvolute to get alpha helix, beta sheet, random coil. It works in solid as well as liquid, strong chemical specificity, and it is a complementary technique. So, if you are like as we discussed in the last slide, we are measuring alpha helix, beta sheet, random coil content in CD. In FTIR also we can get that secondary structural elements present in a quantitative fashion. So, both of these techniques are complementary, right, circular dichromosome and FTIR. You can also do with a lyophilized drug product, right. So, you freeze dried formulation, CD cannot be used. For CD you require a it has to be reconstituted in liquid state, but FTIR can conform it. just you mix with a KBR palette, record the spectrum and you can do that, right. So, you can detect the using secondary structure, and if it detects more beta sheet, that means your the freezing, drying, storage has changed the secondary structural conformation, and that will lead to aggregation risk. Coming back to now Raman spectroscopy, it is a complementary to FTIR as we have seen. It gives you the chemical fingerprint, right, and it is a water friendly. You do not need to worry too much water absorbs or not, because it works on inelastic scattering vibrational information. So, what happens, it can be used in solution, water rich sample, no problem, just we do Raman, Fourier transform Raman spectroscopy. In container, it is a non-destructive technique. So, in a capillary you just put it, shine the laser, do the experiment, you recover back your sample, can be used. It is a minimal water interference, can catalyze through glass, plastic, whatever it is, and it is extremely good in industry. It is good for PAT analysis, process analytical tool analysis it can do, and basically it is a limitation is sometimes you can have a fluorescence background, it is a lower sensitive than IR, therefore, you require a laser. So, if you remember, it is coming from inelastic scattering, it is a feeble radiation. So, lower sensitive than IR for protein. Inline process monitoring PAT tool can be used, can be used for continuous biomanufacturing. Lots of research is going on how to integrate Raman spectroscopy with inline process monitoring. So, Raman probe can monitor nutrient, metabolite, protein concentration that as the reaction is going, and it is a great saver that is coming up in future. So, one can use without sampling, directly from the bioreactor you put a Raman probe and it is reporting about how much nutrients and metabolites are coming, if the protein concentration is going good or not. That is fantastic tool that is developing. So, let us sum up. So, if I want to do protein concentration, UV visible, orthogonal is Raman. If I want to study the folding unfolding, CD, orthogonal is fluorescence. If you want to detect early aggregation, the good technique is fluorescence, you can use CD. If I want to understand the solid state structure, FTIR is a good technique, you can use also Raman. If I want to understand the biocompatibility, CD is technique, orthogonal technique can be FTIR or Raman spectroscopy. And if I want to monitor the process, bioprocess, how the synthesizing by cell, you can use the Raman spectroscopy, and you can also measure using UV visible spectroscopy. So, what I want to emphasize that not one spectroscopic technique is sufficient. It is like using multiple techniques, multiple orthogonal technique, they are all complementary. So, UV visible, quantitative, CD, structure, fluorescence, sensitivity, FTIR, Raman, chemistry. All these techniques are needed when we are dealing with a question in biopharma, right. So, biosimilar or biopharma works, regulator expect all these orthogonal technique to be invoked to answering different questions. So, agreement among the method is more important than going in a depth with one technique. So, just making the or getting the same information using another technique is extremely important, right. So, we will take some case study. Let us take a case study what FDA is suggesting. So, FDA is saying analytics first. You robustly develop a pipeline where your orthogonal biophysical techniques are invoked for doing the comparative analytical assessment CAA, and there are some guidance by FDA which is say October 2025 CES update with reliance of revision 4 that came just recently few days back. It says the flexibility to minimize the clinical burden. Just take a minute. You see, for drug development, how many animals are killed for doing this preclinical or just looking at tox analysis and many of these. If you are making a biosimilar, right, biosimilar is just a carbon copy of already approved drug. Why you have to kill an animal? So, FDA is coming with a regulation that just for showing and looking at the biosimilarity, probably you can do away with a robust analytical technique. You do not need to test this in an animal, and therefore, you can minimize the clinical burden with animal as well as the clinical trial phase 2, phase 3, all you do not need to do it. If your molecules is identical using various orthogonal technique, what we discussed and what we not discussed, if you do that, probably you do not require that extensive clinical trial. So, you minimize clinical burden. If clinical burden is required, the money investment is reduced, and therefore, you can make a cheaper version of like a or with less investment, you can make a robust biosimilar which matches with all parameters with the reference listed drug, right. So, current emphasis is demonstrate no clinically meaningful difference, and with US licensed reference you can do probably with robust analytical assessment with a minimal clinical trials. So, for IgG1 monoclonal antibody biosimilar, the step first is just take UV visible absorption. Question, are we comparing the equivalent material? That is the question we are starting with. So, what experiment we have to design? Take this biosimilar, measure absorption at 280 for the concentration, and then you take the 8 lot of biosimilar, 10 lot of reference product, extinction coefficient verified by amino acid analysis. You can do this robustly. If you are like if you are getting it very comparison, then concentration bias is less than 2%. No abnormal absorption happens because of the aggregation, no turbidity, no aggregation. That shows that your like a biosimilar lot and reference lot is same. Now, why it is important? Because if the quantitation is wrong, every downstream process that you are doing is meaningless. So, early on do it robustly. You emphasize on UV visible baseline equivalence and not similarity of the structure. The outcome is going to be the equivalence compatibility of the dosage, the material is established early on. However, using UV visible you do not have a structural information, we need some other technique to be done, and that technique can start with a far UV CD. So, their question we are asking, is the secondary structure conserved? Now, what we are going to do experiment? Just do the far UV CD, measure the spectra between this 190 to 250, overlay these spectra, all biosimilars and reference lot you can put it. Then to look at the specificity, stress the sample, just generate a stress condition at pH 4, pH 8, or just change the temperature and show that if we do this deliberately your structure differs, right. So, if they are similar, you have an identical minima at 216, 280, which gives you beta C3 IgG signature, and overlapping unfolding curves or melting transition can also be measured. Why it is important? FDA considers secondary structural similarity has a core of HOS higher order second structure requirement. So, that is FDA considers secondary structure is the basic unit that where you can do, and you can demonstrate the global fold of or the backbone hydrogen bonding pattern using CD. So, outcome can be yes, secondary structure is highly similar, tertiary packing or microenvironment can still differ. For that you have to do the near UV CD. So, that is a step third. In a step 3, what we are going to do? Ask the question, are the aromatic side chains and disulfide is the same, have disulfide bond at the same amino acids, or it is a cross disulfide bond has formed. To check that, you require essentially the near UV CD experiment. So, experiment that we are going to do between 250 to 320, we are focusing in this measuring 295 for tryptophan, tyrosine phenylalanine and 262, 275, and lot to lot statistical comparison we are doing. So, result is near identical peak position and intensity we get, no separate distortion under mild stress condition happens, and then we are confident that we are getting the same tertiary structure. Now, FDA reviewer used to detect the subtle packaging difference that may be coming because of different protein production that we have. So, outcome is tertiary structure is comparable, but are like the third question, tertiary structure, this shows tertiary structure robustly comparable. But next question, are there any early dynamics conformation instability? To do that, we need fluorescence. Dynamics changes can be measured by fluorescence. So, are there any early folding or micro environment difference that are there? So, that can be measured by fluorescence. As we know, fluorescence is sensitive to the environment. So, we are measuring the tryptophan fluorescence like exciting at 280, putting emission at 295, and we can also take the stress condition where low level of heat or aggregation or freeze thaw cycle are there. So, if we put it a stress, structure is changed, you can detect using fluorescence. Now, excitation we had 295, emission we are having at 320 and 324. If there is a red shift or any change in the intensity, we can find it out, something is happening, and that ensures that aggregation is happening. If nothing is changing, you will get identical emission spectra. So, CD average the structure across the molecule, but fluorescence detects local unfolding happens before even CD change, and FDA expects orthogonal HOS method. So, no repetition of this same physics that we are doing, you are using fluorescence for looking at the dynamics. So, no hidden tertiary instability or early unfolding is happening, that can be answered by fluorescence. And then next is FTIR, on cell lyophilized product. Suppose your protein is not stable and you want to store in a lyophilized form and then reconstitute it before injection, you can just look at lyophilization freeze thaw cycle is changing or not. So, each solid state structure preserved. To do that, you have to do ATR, FTIR on lyophilized cake, and you get the amide 1, amide 2 structure, you deconvolute, get the alpha helix beta sheet turn. You can put it to humidity stress like a 25 to 75% humidity, and if nothing is changing, you should get the identical secondary structure fraction. If something is changing like on humidity, you will get that something is changing, same humidity induced reversible change can happen, right. So, now CD and fluorescence require solution. FD expects solid state compatibility, that can be coming from FTIR, and FTIR dosage form you can get it. So, solid state structure is also comparable, that can be done by FTIR molecule. Now, finally, biomanufacturing, continuous manufacturing can be measured by Raman spectroscopy in solution or PET relevance. So, here we are measuring each chemical fingerprint consistent and the process is controlled. To do that, we can employ the Raman spectrum fingerprint, Raman spectrum fingerprint. So, we take a formulated drug and we do the multivariant PC analysis from lot to lot, and the cross reference of upstream PET Raman can be invoked. It gives you idea about biosimilar and reference cluster overlap tightly. You can do the PC analysis, you can measure some Euclidean distance, Mann-Levenius distance, all of these you can measure it. So, if there is an outlier, you can easily get it here. So, it is a water tolerant, non-destructive, demonstrate the manufacturing consistency, and FDA is now like just recommending to have a quality by design or PET philosophy, so that you reduce the uncertainty of your drug product production, right. So, manufacturing variability does not introduce hidden difference, that is why you are doing continuous monitoring using Raman spectroscopy, right. So, that is what one can now combine each of these techniques, UV visible, CD, fluorescence, FTIR, Raman, in developing a robust protocol for your drug manufacturing in biopharma industry. Let us stop it here and we do some more case study how we can combine all of these techniques to answer a questions. So, look forward to have you in the next class. Thank you very much.